Hello guys, you're welcome to Slick Academies, and in our last class we discussed glycolysis. Today we shall be discussing further on Krebs cycle, or what some people know as tricarboxylic acid cycle. So for our Krebs cycle to take off successfully, remember in our last class we discussed glycolysis and the last product was pyruvate. But pyruvate cannot be introduced into the tricarboxylic acid cycle, so it has to be converted to another molecule, a 2-carbon molecule. Remember, our pyruvate is a 3-carbon molecule. Now, it has to be converted to a 2-carbon molecule known as acetyl-CoA or acetyl-coenzyme A. And this is facilitated by an enzyme we know as pyruvate dehydrogenase. Now, also here, in the process of converting pyruvate to acetyl-CoA, there's also the conversion of NAD+ to NADH. Then acetyl-CoA goes on to facilitate movement into the Krebs cycle, and the first molecule it comes in contact with is a 4-carbon molecule known as oxaloacetate. It combines with the 2-carbon molecule acetyl-CoA to form a 6-carbon molecule citrate, and this is facilitated by an enzyme known as citrate synthase. The citrate is further rearranged into a different structure, still maintaining its 6-carbon structure, though simply rearranged. That is why it is now called isocitrate. And this rearrangement occurs by loss and gain of water and is facilitated by an enzyme known as aconitase. Now, this isocitrate is now further reduced to a 5-carbon molecule known as alpha ketoglutarate. Now, alpha ketoglutarate is actually a 5-carbon molecule, and this is achieved by the loss of carbon dioxide and the conversion of NAD+ to NADH. And this is facilitated by an enzyme known as isocitrate dehydrogenase. Now, our alpha ketoglutarate, a 5-carbon molecule, is converted to a 4-carbon molecule. But then this molecule gains an extra attachment we call coenzyme A, giving rise to succinyl coenzyme A. It loses the extra carbon by loss of CO2, and this process also involves the conversion of NAD+ to NADH, and it is facilitated by an enzyme known as alpha ketoglutarate dehydrogenase. Now, our succinyl coenzyme A is now converted to succinate. In this case, it goes back to a 4-carbon molecule without the attachment coenzyme A. Here we have the GDP converted to GTP. This is also another source of energy in the body, that is guanosine diphosphate to guanosine triphosphate, which can also give rise to ADP to ATP, that is adenosine diphosphate to adenosine triphosphate. It simply means that at this point, energy is produced. Now, the succinate goes on to be converted to fumarate. Here, the FAD is converted to FADH. Here, FAD simply means flavin adenine dinucleotide. And this process is facilitated by an enzyme known as succinic dehydrogenase. Fumarate is then converted to malate, and it is a process that involves addition of water and facilitated by an enzyme known as fumarase. And the malate goes on to be converted to oxaloacetate. This involves the conversion of NAD+ to NADH, and this process is facilitated by malate dehydrogenase. Now you must have noticed we have gone a full cycle when we got to oxaloacetate, which is a 4-carbon molecule, and another acetyl coenzyme A, which is a 2-carbon molecule, will come in to mix up with oxaloacetate to form a 6-carbon citrate, and the cycle goes on again to come back to oxaloacetate. Now, going back to glycolysis, the net ATP produced is 2, and at the end of it all, one molecule of glucose gives us 2 pyruvates, which also gives rise to 2 acetyl coenzyme A, which now move into the Krebs cycle. And for each cycle in the Krebs cycle, 1 ATP is produced. The process continues as each cycle produces ATP, energy, and also electrons that shall go on into the electron transport chain. Thank you very much for sticking around, and I know you enjoyed today's class. If you have any contributions or questions, please, we'd like to hear from you. And until then, stay blessed.